genetics and evolution. This is quite a long topic, and sorry in advance for any mistakes and mispronunciations I may make. Now, let's just jump into it. Okay, starting off with meiosis is described as a reduction division because the chromosomes are halved from diploid 2N to haploid cells, which has 1N. And the chromosome number is therefore kept constant instead of doubling every generation like mitosis. And this causes genetic variation for gametes, which leads to natural selection, which I'll talk later on in this presentation. In prophase 1, homologous chromosomes pair up to form bivalent in a process called synapsis. And at the end, 4 haploid daughter cells are produced instead of 2. That's the addition of prophase 2, metaphase 2, and anaphase 2, and telophase 2 to this. Let's move on, as this is a brief summary, not a detailed explanation. So the variation is due to crossing over in late prophase 1. The chromosomes tangled up and exchange sections. The chiasmata connects with a non-sister chromatid, so maternal and paternal genes are exchanged. And when these exchanged genes form an offspring, they are said to be recombinant offsprings. And this is also due to independent assortment, which is during metaphase 1, pairs of homologous chromosomes lies independently and randomly at the equator, causing this, as you can see in the diagram here. Now let's move on to gametogenesis in humans. This takes place for males on the left side of the diagram in the tubules of the testes. The germinal epithelial cells divide by mitosis to produce diploid spermatogonia, which then grows and then form the primary spermatocyte. The primary spermatocyte then undergoes meiosis, forming 2 haploid secondary spermatocytes, which continues with meiosis 2, forming 4 spermatids that matures into spermatozoa. And then in females, we have the same, but it's called oogenesis. Well, not the same, it's quite different. It takes place in the ovaries, and the germinal epithelial cell divide by mitosis to produce diploid oogonia. Oogonia starts meiosis and become primary oocytes, which are still diploids. And then, to note, this occurs before a baby is born, a baby girl, that is. And at puberty, the primary oocyte continues to finish meiosis 1 and produce secondary oocyte and first polar body, which is a small haploid cell with less cytoplasm. Each month, 1 secondary oocyte is released into the oviduct to get fertilized. And if fertilization occurs, secondary oocyte undergoes meiosis 2 to form an ovum and a second polar body. Okay, let's move on. Actually, before we move on, so highlighting the main differences, in male it produces spermatocyte, and in female it produces oocyte. And in females, the division of cytoplasm is unequal, while the division of cytoplasm is equal in male. And in female, only 1 gamete is produced, and in male, that's 4 gametes produced. In females, there are polar bodies, but males doesn't have polar bodies produced. Okay, now moving on to gametogenesis in plants. In the male, it takes place in the anther, which is this diagram here. Diploid pollen mother cells divide by meiosis, forming 4 haploid cells. And the nuclei of these cells divide by mitosis, resulting in cells with 2 diploid nuclei. These cells then mature into pollen grains. One of the nuclei is the tube nucleus, and the other is generative nucleus. Now moving on to females. This takes place in the ovules. The diploid spore mater cells divide by meiosis to produce 4 haploid cells. All but one degenerate. This cell develops into an embryo sac. The embryo sac divides by mitosis 3 times, as you can see in the diagram. And they form 8 haploid nuclei. And one of them becomes the female gamete. That's basically all you need to know. Now let's move on. Okay, moving on. A test cross is when you cross a dominant allele organism with a homozygous recessive organism, and the offspring phenotypes can help determine whether the parent is a homozygous or heterozygous dominant. I assume you know what a allele is, a genotype is, a dominant or recessive allele is. Okay, moving on to monohybrid crosses is between two individuals with different alleles at one genetic locus of interest. A locus is a position with which a particular gene is found on a particular chromosome. And then moving on to dihybrid crosses, it deals with the inheritance of two separate genes on different chromosomes at once. Okay, for example, I selected big S and small s for shape and big C and small c for colors. And this is a cross between heterozygous and a homozygous species. This is a heterozygous dominant and a homozygous recessive. Okay, so according to this table, as you can see, it gives the same 4 things 4 times. So the ratio is 4 to 4 to 4 to 4, which simplifies as 1 to 1 to 1 to 1. You can try it yourself. It's more of an experience thing. And to test this observation, you use the chi-square test to test if the expected frequency is equal to the observed frequency. I won't be giving the formula as the formula is given in exams. Just know that the larger sample you use, the higher accuracy it is. And if the chi-squared value is more than the critical value, you reject the null hypothesis. But if it's less than the critical value, you accept the null hypothesis. And another case is when it's crossed between two heterozygous species. It will produce a ratio of 9 to 3 to 3 to 1, but you can work that out by doing it yourself. Now moving on to what an autosomal linkage is. A linkage is the presence of 2 genes on the same chromosome, so that they are inherited together and do not assort independently like most others. This is a very rare case, however, as almost all the links are broken due to crossing over in meiosis. Now, moving on to sex linkage and inheritance. I assume you know that an XX is a female and an XY is a male. So your gender is determined by the father's sperm giving a Y, as the mother always gives the X chromosome. That's a 50-50 chance. A sex linkage is the phenotypic expression of an allele that is dependent on the gender of the individual, as is tied to the sex chromosomes. For example, an X chromosome contains a gene that codes for blood, which is the factor VIII. It has a chance to get the recessive allele, small h, causing the disease hemophilia, which is when the blood can clot. This is a sex-linked gene because it is found on the X chromosome but not on the Y. So it's expressed in male more than the female, as the female needs 2 XH from both. Okay. Moving on to selection and evolution. Starting off with continuous variation, a quantitative difference that has a wide range of phenotypes. When you plot it on a graph, it forms a bell-shaped distribution curve. An example is height and weight. They are not affected by different alleles at a single gene locus. Or if they are affected, they have only small effects. And this continuous variation is a qualitative difference that has clear, distinguishable categories. For example, blood groups, and these are controlled by one or few genes, so alleles on a single gene locus have large effects on it. You can carry out a t-test to determine whether the means of two normally distributed sets of data are different from another. This is kind of similar to the chi-square test, so I won't be going through this. Okay, moving on to natural selection. So before we do that, we have to start on the selection pressure. And these affects the trait favored. For example, body size. Like if your body is small, it might be favored as you might not get hunted so much as people. It can be difficult for other animals to see you. Okay, so there are three types of selection pressure. It's a stabilizing selection, which the average is favored. In the case of body size, it's like average body, not too big, not too small. So narrow the extreme sides, the left and right side of graphs are shortened. So they have a narrower range of characteristics overall. Okay, moving on to directional selection, which is when one extreme is favored. For example, a small body size is favored, so the graph shifts to the left or to the right, depending on where you put it. And the last one, disruptive selection, is when both the extremes are favored. So in the case of body size, it's when the small body size and large body size are favored, so the average dies out, forming this curve right here. Okay, moving on to how you answer the natural selection and the theory of evolution. So you identify the selective pressure, then you identify the selective advantage, and you say the organisms with selective advantage survives, reproducing, and passing on the advantageous alleles to their offsprings. And that makes the allele frequency of advantageous traits increase in the population after many generations, and the disadvantageous allele decrease. Okay, for example, giraffe necks. So giraffes with short necks are dead, as you know, because you can only find giraffes with long neck now. So the selective pressure is the length of the neck, and the selective advantage is those with long neck, so they can reach the leaves on the tree. And the giraffes with long neck survives, reproduces, passing on, yada yada yada. You get the point. And then the theory of evolution is that there's variations among individuals in a species, and those with the strongest and best adapted genes will survive and reproduce in what's known as survival of the fittest. Okay, moving on to speciation. There are pre-zygotic speciations, which is when individuals are physically unable to mate or the gametes are unable to fuse, and post-zygotic speciations, which is when there's a failure of cell division and non-viable offsprings, which means their offspring die too soon. Moving on to the frequently asked in exams, the allopathic speciation. This is when populations are separated from each other geographically, by a physical barrier such as a mountain or a river. So the different habitats they're in, different environments, so different selective pressures, and different advantageous traits, and also different mutations occur. This leads to the genetic drift in the population, which leads to alteration of the allele frequency, causing evolution and a new species form. This is known as the founder's effect. And then there's also sympatric speciation. This is when a new species is evolved from a species that inhabit the same geographic region. And a common way is occurred by polyploidy. Polyploidy is an organism with more than 2 complete sets of chromosomes in its cell. This can happen if the meiosis is abnormal during the gamete reformation, resulting in gamete with 2 sets of chromosomes. And also due to mechanical or behavior abnormalities. There's also selective breeding, which is, for example, in cows, choosing high milk yield cows is choosing individuals with desired traits and breeding them. But farmers avoid inbreeding, because it may cause inbreeding depression, which causes the gene pool to become progressively smaller and weaker over generations. Outbreeding is preferred, so trading with other farms or from the wild. And the Hardy-Weinberg principle is used to calculate the allele genotype and phenotype frequency within a large randomly mating population, with equations like p plus q equals 1 and p squared plus 2pq plus q squared equals 1, with the p representing dominant allele frequency and the q representing recessive allele frequency. But this does not apply when there is a significant selective pressure against the genotype or migration into or out of the population, and also non-random mating. And that's pretty much it for this topic. I might have missed some, but these are most, if not all, of the main things you need to know about this. So if there's any mistake or if you enjoyed, please leave a like, thumbs up, and leave a comment and hit that bell. Thanks for watching.